Welcome to my tutorial. Today we have a topic that is DNA replication. Wow, this is one of the few topics in molecular biology which always students find very typical. Okay, let me explain it a better way. So what DNA are going to be duplicated or replicated, then DNA make what RNA, and then RNA make what protein, and that protein will make you. What the function of protein is actually, the protein have a function that is structure and functional role. But what the function of DNA or gene are involved in the synthesis of protein in the active form. First, this thing are going to be duplicated. This portion, when the parents goes to offspring, so that the genetic material are going to be transferred. That is actually DNA. While this portion, this is called gene expressive portion or gene expression, are going to be here. These are going to be involved in the gene expression. While in some cases they are going to be reverse, which is called reverse transcriptase in the case of HIV. Okay, together this drama, this molecular operation, we call them central diagoma. This is called central diagoma. Central diagoma have a role in our body. I'm talking about a very simple example of that is even the sperm and eggs will form a zygote. What? Zygote are represented, they are going to be the somatics, and that's been germ, they will make zygote. So everything are in the codic form, and so the light of that zygote are going to be forming, and the young one are going to produce. So that is other thing I'm going to discuss. But the everything are in coded form. So lighter the protein are here, they're not specific, they convert into specific. Some will form here, some will form nails, some will form skin. Of course, some will form eyes. So it's very important. So the role of DNA in our body to make protein, and what the protein is, they will make you and me. Wow, what the function of protein? Of course, protein are very important because their structure is critical to its function, like other macromolecule. This is one of the living awesome. This chemistry become biology here. This is a lot of chemistry. DNA will make RNA messenger RNA, and then it make protein. When the DNA goes to the RNA, this process is called transcription. While the protein from RNA, especially messenger RNA, is called translation. This is translation. So then this chemistry become biology, and it will make you, and it will make us. Okay, let me proceed. For replication, there need to be a raw material. For molecular operation, there need to be raw material. So their raw material, which are going to supply energy, that is adenine diphosphate, the oxy we're talking about, guanine diphosphate, cytosine triphosphate, thymine triphosphate. Of course, the phosphate have a very important role. Because when I'm going to represent like that means they are high energy phosphate bond, and then they produce phosphate is going to work, that physiological work, 7 kilocalorie energy going to produce. So phosphate is going to physiological work. With the other raw material, which along with this, that is enzyme. Enzyme is a biocatalyst, so they are agent of life. What sort of enzyme which are going to involve in the replication? That is, for example, that is going to call Helicase. When we talk about S, it's called enzyme. Now that is carase. Now this called ferramase. The other one, which is enormous role, especially in DNA polymerase. The last one, but not the least one, that is called Helicase or Okazaki fragment, because it's been introduced by Okazaki. So these are enzymes which have a role. Along these, this is a third one, the last one, but not the least one, that is single cell binding protein. So these three things together is raw material. They put something on a machine, that machine will be make some something in the palm of another copy, which is called replication. So okay, let me explain the replication in a traditional way. Oh, let's settle this word in a traditional way. Let's going to be start this word in a traditional way. Okay, like traditional way, that is going to be the parent strand that are going to be called daughter strand. Okay, so these three mechanisms through which we are going to mention a replication, but the best one is among those is semi-conservative. But before I'm going to discuss these things, if you know about the direction of these things, that is going to from 5 to 3, from 3 to 5. Why? Because they have a bond which is called phosphatide easterly carrier here, downward, the other is going to be upward. Anyhow, this is very important. And semi-conservative mean 101 new. So traditionally we mention these things in a semi-conservative way. That is semi-conservative way, 1 old, that is old, and that is going to new. While the other one, 75%, they are going to be acceptable. Okay, the other one that is called dispersive, dispersive. This is dispersive. That is hydrogen bond, I will explain these things and promise these things. This is called this one, this is conservative. So that is conservative, the last one, that is going to be dispersive. What? Dispersive, that is going to be dispersive. This is dispersive, that is dispersive. So that is conservative, that is conservative. While this one is dispersive, and this one is semi-conservative. What? Semi-conservative. Okay, let me proceed and settle the bar. We have a double helix structure. We have also one thing you should talk about. We have also mentioned X-ray crystallography. Whenever I'm going to draw the structure like this, one thing which came in my mind, even everybody who teach biology, if he or she know the reality is concerned, the name which is going to be in their mind, the name is Rosalind Franklin. Wow, I'm not sure why she not been shared the Nobel Prize along with Watson, Crick, and Wilton. I don't know why, because we can't say nothing about it. But there's the reality, But anyhow, there's guilty everywhere. So that is a double helix structure. That is B type of DNA. The basic rotation, we have a DNA A type, have more, called more basic is concerned. We call right-hand helix. That is going to discuss Z form. There is the exact manner, and they are thought to be involved in the gene regulation. So okay, so physiologically we are going to mention B type of DNA here. This is going to call hydrogen bond. This is called what? Hydrogen bond. What important things here? First of all, the enzyme which is called Helicase. Helicase will break this one. They will reduce their tension is. So now the strand will be like this. This is going to be Helicase. So Helicase have a very, very important duties being performed. So they break the bond. They break the bond is concerned. Somehow it's been released with another enzyme, Helicase, also going to work to reduce that tension further. But here in the same patient here, I'm talking about these things. They are called single cell binding protein are going to be here. They work on they bind these things. This is if you have a rope and you roll around each other, then you will realize that the structure is going to be. So I said binding protein are going to be here. Let me proceed these things. Then what happened in the next? Then another enzyme which is called Helicase or Kozuki fragment. They're reverse coiling. Reverse coiling. They also were here. The prepase is here. The prepase is going to be here or here. Because they are going to be new strand. That is all discussed about the structure is. These are going to be the new, and that is going to be the old new, that is called in semi-conservative fashion. Then let me draw this thing in the old fashion like that. Again, the better way. Okay, these are going to be the new, they are going to be the new. So this is very important. It's going to be the mature is going to be here. What sort of things that are going to be needed? This is a building unit. What? Building unit. Oh my goodness, this is building unit. This is very important. These are going to be the new strand. How these new strand will form as part of that. But of course, the enzyme have to play a very crucial role. Okay, let me proceed further. Here the building unit, the data revision bond are going to form the guanine cytosine. Way the bond that is was mentioned like that is going to be here. Adenine, the guanine, thymine, and cytosine. They're going to double. There's going to be triple. So, for example, I mentioned guanine with cytosine. There's going to be triple bond. While adenine with thymine, that is going to be double bond. So that is a hydrogen bar going to be here. This is extra. Hypothetically, these things are going to be represented. This is the reality that we are going to mention. These things hypothetically, before Watson and Crick, has been. They were being taught that that's going to be self-replication, but how? The question was that we don't know about how. That time how, but now we know how that is. So what happened in the traditional way? We just consider these things. What happened? The hydrogen bond to the hydrogen bond, the hydrogen bond that is going to be here. The hydrogen bond. They are going to be what they are going to be dissolved. They are going to dissolve the complementary dissolved bases. Hydrogen bond between the bases. They are going to be here. They are bases. Actually they are bases. They are bases. Plus we have mentioned Helicase. They dissolve these things. These are hydrogen bond. I have mentioned here. That is they are going to dissolve. Now what happened? What's going to be the next thing? Each strand, talking about each mean then this is also strand. So each strand have integrity. They will have integrity. That is important. Third step, what each strand is a template. Each strand is a template. So why we call template? It means it's a blueprint. Recall simply is a guide that is that is going to come here. For example, that is 5, then it's going to be 3, then 3, then it's going to be 5. So each strand talking about this is the blueprint, and that is going